Thanks, everybody. Thanks for coming. It's my pleasure to introduce Ruxandra Draghia-Akli today, who I met working through Active. She was actually the co-chair for the Active program, and just very insightful and clear, and just somebody who I looked up to during that crazy time of COVID. But she's had like an amazing career. She did her MD and PhD in Romania, and then spent postdoc in France at the Descartes, and then went to Houston at Baylor, and has like 100 and some odd papers and 100 some odd patents, and then went to Merck for vaccine, and then and is now actually European Commission, the director of the European Commission and deputy director. So just an amazing career, and now is at Johnson & Johnson as head of global health. And today she's going to tell us a bunch of stuff. So
thank you, David. It sounds much better when he presents it. I know I'm going to start with a little bit of a brief introduction of how do I ended up here. It's a winding road. Indeed, I'm Romanian. I went to medical school there. I started a PhD in genetics. And it was just after the fall of the Berlin Wall, won a fellowship and went to Paris to finish my PhD. And serendipity, it was one of the first labs ever doing gene therapy. So I started doing that. And when my PhD was finished, another serendipity, they said, it was for the Europeans, maybe not many here. It was before the Bologna process. So basically, they weren't recognizing diplomas from a country to another. So they said, okay, we love you. We can give you a faculty position, but go to the United States, do a postdoctoral fellowship, and then you can come and be faculty, do research. I arrived at Baylor College of Medicine, and within 10 months, I was faculty, tenure track. They thought that I was doing a very good job. I had the dual appointment in the molecular and cellular biology and in the Center for Cell and Gene Therapy, doing nucleic acid gene therapy and vaccines starting in 1995. So it's like the dinosaur times of gene therapy. Again, serendipity, the experiments went very, very well. And Baylor was very interested in creating a spin-off company around the technology. It operated as a virtual company in my lab for a while. And then it became too big. We had to do GMP manufacturing. So the investors took it as a brick and mortar. Just that the regulators were not ready for cell and gene therapies at the time. And after, putting the first gene therapy out there for an animal application, I was so bored with, not having the opportunity to move faster. I was one of the few experts. I don't know how many of you got your own paper back from Nature to be one of the reviewers on the paper that you just submitted, but it happened to me. I was an expert at the European Commission, and when the position of Director of Health Research, Medical Research was open, they've asked if I'm interesting, and they said, you all are joking. I'm just coming from the American pharma and your government entity. But they said, no, you're going to learn. So it was a really great move. We were able to put into place a lot of public-private partnerships, new funding modalities for research, and really exciting programs, everything from gene therapy and rare diseases, which were where I was coming from, and to vaccines and infectious diseases. So it was really a bubbling initiative. But then, people are saying, yet again, you're going to leave an exciting job where, you can die there on a ginormous salary and whatnot. Why did you leave? And the explanation is, I got a promotion. I was put in charge of all the research and innovation programs, climate change and environment and aeronautics and whatnot. The public-private partnerships were very cool, were extraordinary. I've learned a bunch. The politics was as big as the grants. So I thought that I would need to go back to real life. So I came back to the States. and worked in Merck vaccines until 4 years ago, when I was offered to lead the global public health organization in J&J. So I came to the organization with COVID. And it was from that point on a very interesting journey. So what do we do in global public health in a pharma company, one might wonder? And first I have to disclose all kinds of things, I don't put the kids on, but everything else is there. So this global public health organization is really a non-for-profit therapeutic area within Johnson & Johnson Research and Development that is focusing on major gaps in really addressing the needs of low and middle income countries. So as you all know, about 2 billion people globally don't have access to modern medicine. There is a big gap between when a drug is getting to the market in a high income country and when it's arriving in LMICs. And then when you are looking at the R&D environment, only about 0.2% of all the grants in biomedical research are awarded to investigators that are originating in LMIC. Less than 20% of the clinical trials are conducted in these geographies. And really, 1% of all new drug development investment goes to new drugs for infectious diseases versus others. It's tremendous. We are all struggling with that. So I think that I need to press that. So our purpose in our organization is really to innovate for the world's 2.5 billion people with no access to modern medicine. And we have been relatively successful. We have actually, in a way or another, touched almost 1 billion lives since the inception of the program. We are the #2 research program in what is called the Access to Medicine Index. So this is looking really at research, really targeting the most underfavored populations. And in the last, 4 or 5 years, we were able to secure more than 600 million in external funding, because our organization is a bona fide academic lab. Of course, we are supported very generously by J&J with all the infrastructure and with the salaries of the colleagues, but everything else we need to secure through grants and contracts. So what are we actually working on? So I'll start with the top and with our actually research programs, which basically are focused on MDR-TB, by extension leprosy. We do have quite a vibrant program, and I'm going to focus primarily on that program today, on flaviviruses and dengue more specifically. Corona therapeutics, we have an AMR program, and then we have 2 areas of support to these main therapeutic areas. One, which is really focused on capacity building. So we are talking about capacity building programs per se, so really bringing clinical trial sites in sub-Saharan Africa, for instance, at the same regulatory standards, so they can actually, we can do the trials and submit the data to an FDA or an EMA. We do have, in collaboration with academic investigators in Singapore, at the London School, at the University of Cape Town, Centers for Global Health Discovery. So these are partner discovery centers. And we do have a Center of Pandemic Preparedness and Response. And then on the other side, we really work on bringing digital solution, disease management programs, and data sciences into these spaces. So very cool programs. So for instance, imaging biomarkers for leprosy, or biomarkers for severe dengue in the most affected populations, and all the way to modeling work. And I'm going to present a little bit of that when we are talking about the dengue. So basically, we strive to really deliver this fit for purpose innovation, diversified, but really work with those in the affected geographies. We are partners. We are not going there and actually trying to bring interventions that have been developed for high income countries, but really innovate specifically for these places together with those that are affected. So a little bit historically, the team brought the first new TB drug to market, bedaquiline, after a 40 years gap in innovation in TB. It's called Sirturo. I'm just very happy to say that we just submitted to the FDA our post-marketing commitment 10 years after. Okay, humongous trials following up these individuals for 10 years. We've donated 2 billion doses of Vermox, soil-transmitted helminths. We have an Ebola vaccine, prophylactic, so we were able to undertake during the different Ebola outbreaks clinical trials in the affected populations. I mentioned a little bit about our Centers for Global Health Discovery. We are contributing as Johnson & Johnson, $100 million to the AMR Action Fund. This is an initiative cross-industry bringing together about 10 of the pharma companies that are actually putting the money together in the fund, going to small enterprises or even to academic investigators that actually want to take some of their innovation and bring them as drugs for new antibiotics. And then we are actually contributors to the Africa Frontline Catalytic Fund. So this is basically healthcare workers. We do believe that actually focusing on these diseases can bring the biggest difference to the population. So for instance, taking leprosy, we had a phase 2 proof of concept trial using bedaquiline in leprosy clinics in the Amazon. So you can imagine the complexities of doing that. And now we are moving towards actually phase 3 in India and Brazil, and then possibly in other places, possibly in the United States if we are going as we go. With the latest data from the CDC. So I know that you're going to be very interested in that because we all live on some type of grants and contracts from the different funders. And it isn't a surprise that the US government is the single largest funder of global health R&D. So this is data, specifically we've omitted the COVID period because that is a bit of an anomaly. So this is like the status quo. And you can see that total direct funding across all funders was about $6.1 billion for a 5-year period. Yeah. By comparison, approximately 134 billion for things like oncology and immunology, just to put it into perspective. Also, the industry is coming out of this research, and this is a very worrying trend. You can see actually the dwindling funding when it comes to the industry, and everyone is asking why is industry moving away from infectious diseases? And the reality is that the cost of development are high. They are very similar to any other drug, your oncology or cardiovascular. but your return on investment is absolutely minuscule because you have to go to, or non-existent. Some of these programs that I have mentioned are in fact donation programs. So you are caught into this quandrum where I know that everything that you are doing is so extraordinary because it kind of fuels that innovation all the time without actually in a way compensating for the lack of investment from the pharmaceutical industry side. So a couple of the colleagues asked me who are our funders, and I put here a non-exhaustive list of them. You can see that we do have partnerships throughout the entire discovery, development, and delivery actually path with the regular, suspects, yeah. And during COVID, we've also tried to be very innovative of how to get our collaborators to do the most with that funding, very complementary. So for instance, we worked on with BARDA, we are working on BARDA on a corona treatment. We have collaborated in Europe with the Innovative Medicine Initiative, and we are in the lead of the biggest corona therapeutic program in Europe. Is called CARE, with other pharma and academic partners. Obviously, we participated in Active 1, but also in the WHO Solidarity Plus trial. And we've launched together with BARDA an initiative which is called Blue Night. So basically funding blue sky innovation in areas of interest for BARDA. Everything from, new antivirals and all the way to, Band-Aids that are changing color if underneath you have an infection. Active. Davey mentioned about it, and maybe he can actually share some of the extraordinary stories. I can just say that I truly believe that this was a public-private partnership built for success. Everyone came in really dedicated to make a difference, and it didn't count if you are actually coming from an academic institution or from a regulator or from the industry. Everyone came there to make a difference. We actually screened some more than 800 agents, some novel, some repurposing drugs, 39 agents or combinations who are selected for trials. Actually, at the end, 37 were tested. Molnupiravir and Paxlovid were the two that weren't, because the companies decided to take them and develop them by themselves. 6 showed efficacy, but I think that as important as those 6, negative results actually were as important because they helped combat disinformation and also make sure that you are not giving to patients something that can be detrimental without adding anything. Yeah. So, of course, and I'm not going to dwell onto this, you know very well, both the outpatient and the inpatient master protocols and all the work that is now undertaken. My hope actually for not only Active, but for the clinical trial networks that have been set up during COVID is that they are going to be used in peacetime to bring forward treatments, new antivirals, new antimicrobials. So not everything needs to wait for a new pandemic to be declared. We have so much work, and we have done so much to set up these structures that it would be really, terrible if we would lose them. And, I was looking at an article that was published a few weeks ago from Airfinity. This is one of these research companies that does a lot of statistics and modeling. They were estimating that we have an almost 30% chance in the next 10 years of having another pandemic, similar or bigger than what we had with SARS-CoV-2. 30%. If we don't capitalize on all these learnings, we will be again in that panic and big lagged that we all know. So moving from all that to one of the examples of our antiviral research, I thought that it might be interesting for this particular audience. We are actually working on a pan-serotype anti-dengue small molecule, which we call very dearly 1802. We think that actually focusing on dengue, on other flaviviruses in this time is really quite timely because of the climate change. It is really estimated that by 2080, 60%, 60, of the world population would be in the area endemic for dengue. We've talked with some of the colleagues about asymptomatic disease, and this obviously is a big problem because the vast majority of the infections will be asymptomatic, but you will have actually cases of symptomatic infection, including for those who are getting first-time dengue, and then you can move to, hemorrhagic fever and to the death. And this will only increase in incidence. Very interesting, the dengue serotype are now going through genetic drifts that are leading to new clades and lineages. And unfortunately, the protection, there is no really cross protection, so you can get as many dengues as one might get. And you also can have severe dengue if infected with another serotype. So, it doesn't look pretty, and it is also what we consider the major economic burden. It's incredible to think that the annual global economic burden of dengue is about $9 billion. We've tried a lot of things to actually control both the mosquito and obviously to develop some vaccines with relative more or less success. And we do see our small molecule antiviral to be used for prevention and or the treatment of dengue as one part of the solution, together with vector control, obviously the diagnostics, and the vaccines. Probably some of you who are working in this space know that while there are a couple of vaccines for dengue, they actually are not very good because they are unbalanced. They can actually give a lot of complications. And the Demaxia, so the Sanofi vaccine, actually has a very limited label. When given to naive populations, unfortunately, it can result in death if one encounters one of the serotypes. So, what do we do? Here is a little bit of the preclinical data with 1802. We have actually seen that this small molecule antiviral is covering very well all the serotypes and the genotypes of dengue. We have actually tested it in vivo in mice for all the serotypes in non-human primates. And you can see here in non-human primates is a panel. I don't think that this is, oh, it does. here. That basically here is the classical, viral load. It is basically controlling completely in a prophylactic manner the infection. We have actually undertook quite a large number of studies, including a single ascending dose, showing really very good pharmacokinetics and a very long half-life, which I'm going to show in a couple of slides, is interesting when we are thinking about the community prophylactic solutions. So basically, terminal half-life somewhere between 6.3 and 9.2 days. Safety, no significant adverse effects, no clinical relevant changes in all kinds of stuff. We have sometimes rush. In this particular study, we had these two cases. They did not replicate in any other subsequent studies. And here is kind of like the preview of what we are going to soon publish, but I thought that it might be interesting. So we undertook a study, human challenge study with dengue-free, and we've selected dengue-free specifically because our compound is the least active against dengue-free. So we wanted to put ourselves in the most stringent conditions. This particular study was done in collaboration with Anna Durbin at Johns Hopkins and at the University of Vermont. So basically the study, we had placebo, of course. We have our high dose, so loading period of 5 days followed by an daily administration of the antiviral for another 21 days, and then follow up the individual for 84 days per the recommendation of our regulators. We also currently are undertaking the low and middle dose, and do actually also plan for another part 2. Basically, all this first part was in unfed conditions. In part 2, we will actually do different formulations of the drug and in fed conditions to really identify the most appropriate dosage regimen for our phase 3. So here are the results. They are quite spectacular for an antiviral. So basically, our primary endpoint was the area under the curve for our RNA viral load. And you can see that we actually have a tremendous difference between the placebo and the treated group. To make a very long story short, 6 out of 10 individuals that were actually treated showed complete absence of the RNA, 0 in the control group. Very interesting. We are talking about climate change. So, and we are talking about Johns Hopkins and Vermont. So you know where the geography is. So individuals were screened for inclusion into the clinical trial, and dosing started about 7 days later. We had one individual that had a very strange IgG-IgM profile, and when we've looked a little bit closer, it turned out that in that 7 days, that individual was infected with San Luis encephalitis, that they've got, it's in the same family with dengue. It's another flavivirus. It's cross-reactive with dengue. So they've got it in Maryland, within 7 days. What's the likelihood? It means that there are much more out there, and we basically underdiagnose enormously. And then 7 out of 9 individuals has no signs and symptoms. In parallel, we are actually moving to a field study. So we wanted to see if actually this is holding true in a population that typically sees all dengue serotypes and sometimes actually these new genotypes. So basically, this is the a community-based phase 2, phase 3 study. It's a little bit the same type of design like in your ring vaccination, but applied for prophylaxis. So basically, we are identifying the dengue infective index case. Yeah, they typically come for diagnosis, and we are actually enrolling into the clinical trial all their household contacts. In these populations, they typically are many, and we are randomizing the household contacts to either low dose active drug, high dose active drug, or placebo. And then basically, if you're thinking about it, in those household contacts, you're going to have individuals that are completely naive, so they have not had the opportunity to be bit by the mosquito. So that would be the equivalent of the pre-exposure prophylaxis. There are going to be, household contacts that might have been bitten by the mosquito, but they are in those 7 days of incubation. So that would be the equivalent of your post-exposure prophylaxis. And then obviously we have a long list of secondary objectives, including work on biomarkers, trying to see, can we identify individuals that would eventually develop severe dengue versus the others. It's an interesting study also because most clinical trial sites are not used with community-based trials. So to start that trial costed us about 2 years of training of these sites to bring them up to speed to be able to implement this particular design. We were helped actually by our modeling work. So we really developed in collaboration with MIT and with Harvard modeling tools. So we can, with about 85% accuracy, predict that an outbreak would occur in a specific location about 6 to 8 weeks before the outbreak is happening, which means that we could actually be at that particular clinical trial site and be prepared when the outbreak is happening. We also think that this particular, and we've actually kind of looked at the model and asked, could that model have predicted past outbreaks in the different locations and geographies? And indeed, it can. So it's really very exciting to do that. We have all the tools, so we said, why don't we apply them to the other flaviviruses? So we started a program very recently on looking actually at maybe designing, maybe finding some new antivirals that would have a broad spectrum and address more than one single flavivirus. And coming back to, one of the points that I've made at the beginning of everything is about collaboration, partnerships, and money. Yeah. This is a little bit how it works. So basically, we do have grants with our colleagues from Belgium, because many of the members of the team are based there, for very basic sciences in the flavivirus program. We are part of one of the AVID grants together with the University of North Carolina to look on more structural biology, biochemical assays, phenotypic high throughput screening. And then we do have a big collaboration with Duke NUS in Singapore. They do have really a large collection of every flyby-virus on the planet and every subtype and everything else, and animal models. And indeed, we are actually working on them primarily for the in vivo studies and other. So it's basically a large global collaboration to see, can we actually beat these viruses? And you can see here just an example. Probably many of you are working in this space of drug discovery and development. It is really an enormous effort, which we think that now is a moment as a community, as we have talked about a little bit about Active, to really capitalize on everything that we've learned and take it forward. So with this, I really would like to end. When we go in and talk to the communities, the first thing that we hear is whatever you are doing is life-saving. And I think that whatever you are doing is life-saving. And we really, really, really need to focus on these communities in low and middle income countries, because we are in fact their champions. So thank you. And if you have questions, I'll be happy to.
Time for a few questions, right? Good.
I guess I had a question about your capacity building in these low and middle income countries. Are you engaging with local researchers there, or do you have partnerships with local universities? How does that work?
So we do have a number of activities. We do actually have a fellowship program. So we do bring, in particular, medical doctors, sometimes PhDs doing epidemiology in these countries. And we train them in pharmaceutical development, regulatory sciences, sometimes epidemiology. A couple of them got a master's in public health, and all that with the goal of them going back to their home country and putting in place, actually bringing up to speed their clinical trial sites so they can be then included in these bigger networks. We also work together with EDCTP, if you know, so the European Developing Countries Clinical Trial Partnership is a mouthful, and other pharma to actually network clinical trial networks in sub-Saharan Africa, and again, organize courses, particularly in regulatory sciences. And then we've done something that is like the Antichrist, which is we took all the clinical trial SOPs that we run in J&J. There were 680 of them. And we created a little group in our group looking at every single one of them and ask which are essential to get quality data that can be submitted to stringent regulatory authority. And we've actually identified 52 categories, 168 actually essential SOPs. So we actually are going to the sites and training them in those essential SOPs. And some are, you know, very basic. Is like, how do you qualify a fridge? And all the way to, you know, much more complicated, or, you know, how do you follow up the patients?
Yeah, wow. That is not usual, but laudable for sure. Anybody online with any questions? A lot of people. Good to see. Okay, so
I have a question.
yes, go for it.
I was just wondering if you could talk, sorry, I'm sure this topic is above my head, but just kind of in thinking about the dengue virus project or other projects that you're working on, how you think at this stage about kind of accessibility to the populations that need it most. I know that was very relevant for bedaquiline recently and kind of how you take the competing interests of the kind of pharmaceutical company with access to medications.
Yeah, so the story of bedaquiline was not really represented correctly in the press, because the vast majority of bedaquiline is actually procured by the Global Fund and not by the countries directly. And actually, Global Fund has a very low preferential price. Also, bedaquiline came off patent in July. So, the generic manufacturers can take a stab to it. For the dengue small molecule, I think that first is a small molecule. So the manufacturing costs and whatnot probably will be more reasonable. The cold chain is not like the vaccines, is not like the monoclonal antibodies, so it's going to be more reasonable. And we are actually working with the communities to see how we can actually bring the drug to them. Very interestingly, we do believe that if we are using it in a prophylactic setting, the individuals will have to take it within just the 30 days of an outbreak. A dengue outbreak lasts 30 days. And on these drugs, you will diminish dramatically transmission. Actually, we have very cute data in the mosquitoes. If they are fed on the blood of an individual that gets the drug, they cannot transmit the disease anymore. Cool. So, there are all these things that are going to, but there is some road for the access and the portability.
So I've been advised by a lot of people never to start a diagnostics company, and I noticed that in the minimal infectious disease research dollar pipeline, that it seems like most is going to drug development and vaccine and other development, and a lot less goes to diagnostics. I also didn't see anything on your slides about diagnostics. I was wondering what percentage of work that you're overseeing is diagnostics work.
So we do actually work in developing point of care diagnostics for dengue. We do actually work. So for instance, for leprosy, we are working on not only imaging biomarkers, but also blood biomarkers for more accurate diagnosis. Now leprosy is diagnosed by a mouse foot path assay. So basically, you inject the piece of biopsy in the foot path of a mouse, and you look at that mouse for a year. That's the diagnostics, and is also how you assess treatment. So we are working on all that. It's a little bit earlier, but for TB, we are actually donating the machines, and we are working with a diagnostic company to actually produce the little kits that would help us with really assessing at the point of care multi-drug resistance. So we do all that, but it has to be really fitting within our program. There is also FIND. Maybe you're familiar with the diagnostic work there, and we do work with them on some of the programs.
But you're not going to get rich.
No, yeah, I've been told it's hard to make one profitable.
You can always work for Biomérieux or for. I think that there is a hand up there.
Okay, Steve.
Yeah, hi. I work on dengue with CDC, so it's great to hear about the progress with this new antiviral 1802. And I just wondered if you have planned hospital studies to treat hospitalized patients with dengue with this compound.
Yes, we actually did start a study in Singapore. Unfortunately, Mother Nature was not on our side, because we started the study and then during COVID. And it was extremely difficult. We had to actually stop the study. We were in this conflicting period where individuals are told if they have a fever to stay home. And we really need to start treatment within the first,
2-3 days of the signs and symptoms. So what we do plan now, we've paused the study. We are going to actually have an interim evaluation of our field study. And we would like to also undertake an additional human challenge study, and then with all that data, go back to a treatment study.
Good, thanks.
Any other questions online? Okay,
Thank
thank you very much.